Okay, good day, class. Professor Lonick here. Take two. Uh, I'm. I've got some new technology here, my microphone, and hopefully you'll be able to hear me. These movies will only get better. Okay, so this is the diversity of microbial life. This is part one, and then I will stop just at viruses and continue the second part of the presentation in a day or so. You'll see it posted. So with this week's lesson, we're going to look at some of the microbes that make up the diverse world of microbiology. And in order to continue or to start, let's go over some review of phylogeny and taxonomy. So again, phylogeny means who's related to who, or the relatedness of organisms to each other. And again, we base it on the nucleotide sequences of what genes? Prokaryotes, the ribosomal 16S RNA. And for eukaryotes, it's a different gene. It's the 18S rRNA. So in order to really appreciate diversity, we've got to know where it started from. So this is a phylogenetic tree, or the tree of life, and we know that at one point there was a common ancestor to all living things. That is the root of the tree. And then from there, over time, organisms began to diversify. And we now have many groups with many features. And again, through this technology, we are able, gene sequencing, to place the organisms in that timeline, or in their relationship to each other. Taxonomy, again, is the classification of organisms and also the naming of them. So we start with a domain, and there are 3 domains listed there. And in this group, it's a very general category, a lot of organisms with a few traits in common. And those few traits are what make up the domain or domains. As you go from domain to your largest general groups, you will travel to kingdoms, and then phylum, class, order, family, genus, and species. So as you go from larger, more general groups down through the taxonomy levels, you will end up finally with a species where the organisms, there are a few of them, but they all have the same feature or features. If you look at this grid here, you'll notice that only Eukarya have kingdoms, or use the kingdoms. Bacteria and archaea jump right over that. They go from domain, and then they start with phyla or phylum. Another thing you'll notice about bacteria, way at the end near the species, for eukaryotes, that is the last group. For bacteria, though, there may be subgroups known as strains. So a bacteria may belong to a species, but again, there is just one little feature from that species that may differ, and it will make it a strain. For instance, we have Escherichia coli. The species is a very friendly guy, and they live in our gut. And in fact, they are beneficial to us. But then there are some strains of that Escherichia coli that produce toxins. And in that case, that is what makes it pathogenic, or a dangerous strain of Escherichia coli. Okay, now as far as naming, taxonomy uses two names in naming a microorganism, and they use the genus and the species. So those are the bottom two parts of the taxonomical categorizing. The genus is usually a noun, and it's your first word of the scientific name. And the species is the second word. And the species name is usually a characteristic or a word that describes a bacterium. Again, I'm talking about bacteria here, but describes the organism in general. Now, there are rules for writing the scientific names, and the rule is the genus is always the first word, and the first letter is capitalized, and the species is the second word, and the first letter of that name is always lowercase. Another rule is that both names are in italics. Got to give them a little respect. And then finally, you may abbreviate for the scientific name, but that's only after you've written the full name out first. So, for example, if you're writing about the bacteria Staphylococcus aureus, you would first have to write out the entire name, full name of that organism, Staphylococcus aureus. If you refer to that organism later on in the paragraph or in the writing, then you may abbreviate it as S. aureus. Now, why is that so important? And I'm going to tell you, whether you want to hear it or not. But I'm going to tell you that, if you don't write out that full name initially, you aren't really specifically telling your audience what that organism is. And this is what I'm going to show you. Okay, this is a headline from the New York Times. Back in 1999, we had, I'm sure some of you may remember, we had an E. coli breakout, one of those bad strains of Escherichia coli, breakout at the Washington County Fairgrounds. Several people did die. So it was a pretty horrific outbreak. So this is the headline talking about that, or the title, "Deadly Germ Taints a Tradition. E. coli devastates families and leaves a fair in doubt." So if you look at that E. coli word in the title, you may assume that it's Escherichia coli, but in fact, we don't know for certain. It could have been, given your choices, the guy at the fairgrounds, or a protozoa named Entamoeba coli. So there is a protist that can has the same abbreviation, or the bacterium Escherichia coli. So that's why it's important to completely write out the scientific name of an organism so that there is no confusion initially. Okay, a little more on taxonomy and phylogeny. This is a review or repeat from, I think, my first lecture. They both, taxonomy and phylogeny, have grown, have evolved over time. And again, we started out with Carolus Linnaeus with the 2 kingdoms up to Whitaker in 1969 with 5 kingdoms. Again, even beyond that, in 1977, Carl Woese determined or proposed that there are domains, 3 of them, that are even above and beyond the kingdoms. And again, those are the bacteria, archaea, and eukarya. So as time goes on and we are learning more, and through the technology or through research, we are constantly adapting are adapting the information. So now I'm going to get into the groups of these three quote-unquote domains. General information, the phyla of the bacteria, and that's because these are your microbes. We've got, I think, 5 common groups or major groups here. There are more, but these groups we find in and on us. The Firmicutes is a phyla, and that is your gram-positive bacteria. They've got the gram-positive cell wall makeup. They also get that name from the gram stain that you're going to be doing probably this week or next, that determines their cell wall makeup. Another phyla is the Actinobacteria, and that is gram-positive, a little different though than from your Firmicutes. Proteobacteria, this is a phyla or phylum of gram-negative, mostly bacilli, and they're made up of several subgroups there. We've got them listed. Interesting enough, the alpha subgroup, they find, or there's been information or data that suggests that from that group of proteobacteria, the mitochondria evolved. We have Bacteroidetes, which are gram-negatives, and these guys, many of them in a healthy gut, many of them make up the gut microbiome, and they are important for fermentation, also important to keep our immune system healthy. And then finally, that phylum of Cyanobacteria. These are your photosynthetic, they photosynthesize bacteria. And from this phylum, the chloroplasts seem to have evolved from. So that's some general information of the microbe bacteria group. Now again, the tree, the phylogenetic tree is always evolving, always growing, and up until about the 1990s, they thought there were only 3 domains. But now in about 2013, 2016, they propose that there actually is a subdomain of bacteria known as the candidate phyla radiation group. And these bacteria are nano-sized. They are much smaller than the average bacterial cell. They don't have as many genes as average bacterial cell, and they cannot be cultured, cannot be grown or isolated and grown in a lab. How they found them was really by doing metagenomics, which in other words, they took a sample from an environment, and they were able to take that sample and analyze all the genes in that sample. And this is where they discovered there actually are another group of microbes that make up another part of the bacterial domain. And they are a very diverse group. They make up of 15% of all biodiversity. They cannot be linked into doing just one thing. But what they have found is that they cannot be cultured. They really need to, they are able to live only in communities, only in the presence of other microbes. They also have discovered that in the archaea domain, there is a subgroup DPAN, and that is an acronym for five of the phyla that they have named in that group. And also the Asgard, which is kind of almost the link between the archaea and Euchera. So I'm going to just talk to you about some of the DPAN. Okay, the DPAN can be found in groundwater and in other environments. They do seem, though, like I said, to belong to the archaea group. At some point, though, there's that Asgard group, which they found, and the microbes in that group are very closely related to Euchera. So it is proposed that the Euchera develop from the Asgard group. So this is where we are finding more and more each day. At one point, it was thought that the archaea are closely related to bacteria, but now in fact we're seeing that it almost is that missing link. The archaea have evolved from the bacteria, but that it's actually more of a missing link and closer related to the eukaryotic cells. So let me, okay, some of the things with CPR is that they are small genomes. Again, I said they're ultra small and very abundant, but live in communities. They are obligate symbionts, which means that they need the community. They need other bacteria, other microbes in order to sustain their life. And the thought is that maybe these other bacteria produce metabolites that they use, or that have enzymes that carry on metabolic processes that maybe some of these CPR cannot do. They are very limited in their biosynthetic capabilities. So in other words, like I said, they cannot do full screen metabolism like your bacteria can. Okay. So we're finding more out every day. They have found, though, that again, like I said, the CPRs, they're unable to be grown, isolated and grown, except that they have found one group, the TM7s. They were able to grow that in the presence of another specific microorganism. And this TM7 has been linked to periodontal disease in humans. Okay. Interesting enough, this TM7 CPR from the CPR group has been found in dolphin mouths, and certain CPR bacteria was found in some of the blood of human subjects. So this is kind of like the dark page of microbiology. More will be found out as we get better at growing it and researching these subgroups. Okay, this is the biology. This is the microbes. This is the bacteria, specifically I'm talking about for microbes. And again, we know the importance of microbes, specifically bacteria, to us. On the left there are some of those facts we talked about a little earlier, I think in PowerPoint 1. And again, the percentage of humans having the same microbiome is only about 20%, 10 to 20%. Your microbiome is going to be most likely 80 to 90% different than from your neighbor or your sister or husband's microbiome. And this is affected by environment, by what we eat, illness, where we live. Whereas our genes from between each other, our genes are about 99% 0.9% the same. Another interesting fact is that there are 10 times as many bacteria outside of us on our bodies as there are human cells in the body. So that's some interesting facts. There are, I want to say though, with the human microbiome, we don't have the same phyla of bacteria living in every part of our body. So we've got, my face is blocking the phyla of bacteria, but each color represents a different phyla of bacteria. And if you look at the ear canal, we've got a large proportion of brown, which I believe is the, I forget which phyla, but it is, Firmicutes maybe. But if you look at that, it's just saying generally that different parts of the body have different microbiomes, okay? And this again is taken average from healthy individuals. The rule is when you get further down into within us, like the small intestine or large intestine, you're going to get phyla of bacteria that really don't need or grow in the presence of oxygen. So in the large intestine there, those I think are yellow. I think they're mostly proteobacteria, or there's some other phyla that is gram negative. They're actually anaerobic. They don't need oxygen. So again, it's also the conditions in the body. It's the part of the body due to the environmental conditions at that part that will promote the growth of certain phyla. Okay, healthy growth of phyla. Okay, the microbes that are eukaryotes. Okay, again, these eukaryotic cells have nucleus, a cell membrane around the DNA. They also contain cell organelles and a membranous system such as endoplasmic reticulum, which you do not find in bacterial cells. We've got two groups. We've got the macro, so the big ones you see, you can physically see, and then the micro, which is what we're talking about. And these include protozoa and algae, the protist, and some fungi. And the following words are examples of protozoa. And we've got ciliates, the amoebas. The apicomplexans are parasitic protists that live within blood cells. They need to live within our red blood cells or white blood cells. So again, that is a little blurb about the eukaryotic microbes. Another group that makes up our microbiome are the yeast and fungi. That's why you see the MYCO, myco. That means mycology or feast. That means fungi, molds, yeast. Again, they are part of our human microbiome. And if you look at different areas, the oral cavity, the lungs, gastrointestinal, and skin, we've got different species of those that live in those different areas. The ones in the red letters are the ones that could potentially cause disease, that can be pathogenic. And this usually they're opportunistic. So if an individual's microbiome is compromised and they're open to infection, these microorganisms will then cause disease. Okay, and of course, let me see. We see oral cavity, candida, lungs, candida. So there's that yeast that can cause. Normally, it doesn't, but it can may be present, but can cause disease. And in the oral cavity, candida can cause thrush if the potential environment is compromised. Okay, so we do have those yeast and fungi living within us too. So anyhow, I'm going to end here. Where do viruses fit in? And we'll talk more about that in my second presentation. I hope you all are doing well. Again, as a reminder, there's going to be an exam due Friday. I will post it probably tomorrow and hope to get the second presentation up there by tomorrow. And again, if you have any questions or want to go over something, please don't hesitate to contact me. And as an FYI, I will not be having office hours today. This is Monday, February 15th. I've been called into work, so I won't have the office hours today, but I will have them on Wednesday from 12 to 1. Okay? I hope you all enjoy the week. Stay posted with the snow again. It's sounding like we may get a couple storms, but look at the website, the school website, for more information, and we'll work something out if school is canceled this week. Anyhow, all right, have a good week, and hope to see you soon. Bye, everybody.